Sorry, I'm a bit late. Okay. So let's start our lecture. Let's start with Al-Fatiha first. Okay. And your own prayer. Okay. All right. Okay, so let's continue with lecture chapter 3. So we are going to have our lecture for 2 hours today. Okay. We've done for your class, we've stopped at osmosis. Okay. So let's continue with osmosis. So basically, um, before osmosis, we have looked at, for example, the different types of the different functions of membrane proteins. Okay, so there is 6 different function of membrane proteins. So some will function as transport protein to transport substances across membrane, either in or out. Okay, so there's 2 types. We have the channel protein or pore protein, such as aquaporins, and then we have the carrier proteins. So carrier protein, it has a binding site to bind to the solutes. Okay, so every carrier or channel protein, it is specific for the solute that it is going to move. Okay. So for the carrier protein, it only involved in active transport. Sorry, for carrier protein, it involved in both active and passive transport. For channel protein, it involved only in passive transport. Okay, so if carrier protein involved in active transport, it requires energy, such as from example ATP. Okay, so and then we have transport protein that function as enzymes. Okay, transport protein that involved in signal transduction, where this sort of protein, we call it as receptor protein. So where it has binding site to bind to the signaling molecule. So example of signaling molecule can be hormone. Okay, so when hormone, for example, a certain hormone binds to this receptor protein, it causes the receptor protein to change its shape. Okay. So once the protein changes its shape, for example, it activates the protein inside cell. So for example, this protein inside the cell can be enzyme. So this causes the enzyme to be activated and causes, for example, a series of other reaction to occur inside cell. Okay. So it relays information, or it relays message into the cell. It means that for signal transduction, it causes a series of reaction to occur inside cells. Okay, signal transduction. And then we have membrane protein that function in cell recognition. So where it involves glycoprotein. Okay, glycoprotein, such as, for example, we have here certain structure of carbohydrates, short chains of carbohydrate binds to the protein to form glycoprotein that function as, for example, antigen. Okay, or cell tag. Okay, cell. So this one is cell recognition. It identifies cells that are of the same type, so that it forms tissue. From tissue, it forms organ. Okay. And then, so that is glycoprotein, same goes to the glycolipids lah. Okay. So, and then we have here the proteins that involve in intercellular joining, intercellular joining, such as the tight junction, the gap junction. So it joins cell together. And then we have here the protein that attached to the ECM and also to the cytoskeleton, attached to the CM on the outside of the cell. Okay, so and then attached to the cytoskeleton on inside of the cell. So this one, it applies for animal cell. Okay, so animal cell, it has no cell wall. So in order to maintain the shape of the cell, to maintain the plasma membrane, so the embedded protein, that the transmembrane protein, will be attached to the ECM. Okay. So we also looked at the concept of simple diffusion. Okay, so that one. Okay, and then now we are going to look at the concept of osmosis. Okay. So osmosis is also part of passive transport. So passive transport, there's three that we are going to look at. We have done on the simple diffusion. So there's the osmosis and also facilitated diffusion. Okay, so let's look at on the osmosis. Okay, passive transport is basically when solutes or molecule move down its concentration gradient from high to low. Okay, so this one applies for the movement of water. Okay, so osmosis. So osmosis, it involves the movement of water from its high water free water molecule to lower free water molecule area. Okay, so it says here. Okay, so the osmosis, the diffusion of water molecules across a selectively permeable membrane. Okay, so it means that this membrane allows only for the movement of water, right? So a selectively permeable membrane from the solution of lower solute concentration. This side it has lower solute concentration, has a lot of water molecule that are free to move. Okay, to a solution of higher solute concentration. So this area it has more solute. For example, this side here can have a lot of sugar dissolved inside. So when there's a lot of sugar dissolved inside, there will be less water molecule, less free water molecule. Why do I say free? Because the rest of the water molecule will involve in the formation of hydration shell to cause the sugar to be able to dissolve in water. Okay, so this side here it has higher solute concentration with lower free water molecule. Okay, so until the solute concentration on both sides of the membrane is equal, so the water will move from from this side here to this side here. Okay, from lower solute concentration to higher solute concentration. Okay, so let's look at this U tube here. Okay, so this U-shaped tube. Okay, U-shaped tube. So this U-shaped tube, it is divided into two regions. It is divided with the presence of selectively permeable membrane in between the two solutions. Okay, so let's say for example on this side here, we label this side as A. Okay, this side is B. Okay, so on the A side, as you can see, it has lower concentration of sugar. Okay, lower concentration of sugar, it means that on this side here it has higher or more, okay, free water molecules. Okay, lowest concentration of solute, in this case sugar. Okay, so this region here, the A region here, it has more free water molecule. On B side, okay, on B side it has more solute, higher solute concentration. So when the B side has more concentration of solute, it means that it has less and less. Saya confused lah. Less, okay, less ini sebab more lah. More, less. Okay, less free water molecule. Okay, so higher solute with less free water molecule. Okay, so this is at the beginning of the experiment, initial. So after you leave this tube for a while, you will see that on the B side, the level of the solution will increase. Okay, on the A side, the level of the solution will decrease. Okay, so why? Because there will be movement of water from A to B. Okay, so so it says here, a U-shaped glass tube with a selectively permeable membrane separating two sugar solution. Okay, so the membrane is selectively permeable. So you will see that water move from an area of higher, okay, sorry, higher to lower, okay, free water concentration, or lower to higher solute concentration. Okay, siapa yang tak mute tu tolong mute ya. I can hear noises. Okay. Muhammad Adib, can you please silent mute yourself?
Hmm.
Okay. Apa boleh nak? Okay. Okay, so next one. Okay. So it says here, water molecules can pass through pores, but sugar molecule cannot. Cannot. So the semi-permeable membrane will only allow for the movement of water molecule, right? So as you can see from this animation, water will move from lower solute concentration to higher solute concentration. Itu salah satu cara yang awak nak explain. Ataupun water move from higher free water concentration to lower free water concentration, this statement here. Okay. So that is for the process of osmosis. Okay, so this side has more solute molecule and fewer water molecule. As you can see, for the area that has higher solute concentration, it has less free water molecule. Why? Because the rest of the water molecule will involve in the formation of hydration shell. Okay, as you can see. Okay. So okay, let's look at on the concept of tonicity. Okay, so tonicity is the ability of a solution surrounding a cell to cause that cell to gain or lose water. Okay, so this is the definition of tonicity. The ability of a solution surrounding a cell. So this is a cell. So this cell is in a solution. So the solution can be isotonic. So isotonic, what is isotonic solution? The solution having a concentration that is the same as inside of that cell. Okay. So we have hypertonic solution and hypotonic solution. So for this first one, isotonic solution. So the solution solute concentration is the same as inside the cell. Okay, same, iso. So it says here no net movement of water. When it says here no net movement of water, it doesn't mean that the water stop moving. Okay, so it means that water move in and out of the cell at the same rate. In and out, water still move but at the same rate. That is what it meant by no net water movement. Okay. So because water move in and out at the same rate, so therefore the water will stay the same size. Okay. So and then for the second one, hypertonic solution. So hypertonic solution, the solution concentration here, the solute concentration is greater than inside of the cell. So there will be more solute outside compared to inside. So what happens is that water from inside the cell will move out. Okay, the movement of water is through the process of osmosis. Whenever you are describing the movement of water, it is always involve the process of osmosis. Okay, so water loses water. Cell loses water. So and then for the hypotonic solution, okay. So for hypotonic solution, its solute concentration is less than inside of the water. Okay, less than inside of the cell. Okay. So less solute outside, more solute inside cell. So it causes the water from the hypotonic solution to enter into the cell. So cell gains water. Water move into the cell by the process of osmosis. Okay. So water move from lower solute concentration to higher solute concentration. Water move from lower solute concentration to higher solute concentration. But this one here, for isotonic, the water move at the same rates in and out of the cell. So let's look at this diagram here. So this diagram here, this slide, it shows you for if you immerse animal cell. Okay, so this one it applies for animal cell. So highlight that. This one you put a title here, animal cell. Okay, so when you place the animal cell in hypotonic solution, okay, hypotonic solution, there will be more water outside cell. So the water will enter into the cell through the process of osmosis. Okay, water will enter the cell faster than it leaves the cell. So as water move into the cell through the process of osmosis, because this is animal cell, it doesn't have any cell wall, it causes the cell to get bigger, bigger and bigger. So it will swell and then it will eventually burst, or the term is lyse. Okay, burst or lyse. And then, but if you place the
Macam
extra cell, okay,
kalau kata explode boleh tak?
apa dia? Ha?
Kalau kata explode,
Suara putus-putus lah.
kalau kata explode boleh tak?
Explode. Ekstrim sangat. Macam bom. Burst lah. Boleh?
Ya, boleh. Boleh. Mungkin ada.
Okay. Alright. Burst. Okay. So and then bukan saya tak kasi awak guna terms lain, tapi kalau boleh awak guna burst. Okay, so explode boleh lah juga kan. So it burst. Okay. So and then okay, so next one is if you immerse the animal cell in a solution that is isotonic. Okay, concentration of solute outside and inside the cell is the same. Okay, so there will be no net movement of water across the plasma membrane. Again, no net movement means that the water diffuse into the cell and the water diffuse out of the cell at the same rate. Okay, same amount, same rate. So water flows across the membrane at the same rate. Kan, no net movement, same rate. So highlight kat sini. So because the water move in and out of the cell at the same rate, it maintains the volume of the cell, ISO. Okay, so next, if you immerse the animal cell, the red blood cells, in hypertonic solution, it causes water to diffuse out, okay, out of the cell. Cell loses water through the process of osmosis. Okay, so it causes the cell to become shriveled and then die. Please do not says straight away the cell died. You have to describe what causes the cell to die, because water diffuse out and causes the cell to shrivel and then die. Probably die. Doesn't necessarily die. It can die. Okay. So and then example effects of increasing salinity to lake lake to increasing salinity of lake to animal. Okay, so animal that lives in aquatic environment that is really saline, apa tasik yang very saline ataupun kat laut lah paling senang kan. So they have mechanism to adapt to the situation. Kan, osmoregulation we call it. Sekejap kelas. Okay. Okay, so for animal cell, the normal solution for animal cell to be in is in isotonic solution. Okay, so especially those that live on land lah. Yeah, so those that live on land such as us human. So the normal the normal solution for animal cell to be in is in isotonic surrounding. Okay. But for plant cell, okay, so for plant cell, as you can see, so if you immerse the plant cell in 3 different type of solution. So for the first one, if you immerse, if the plant cell is in hypotonic solution, like the one that we have done in the experiment 2, and in experiment 2, you apply water to the leaf, okay? So you will see that the water will enter into the cell, and then the excess water will be stored in the in the central vacuole, kan? So because water keep on moving into the cell through the process of osmosis, and because the plant cell is surrounded by cell wall, kan? So the plant cell will not burst, okay? But it will become turgid, kan? So turgid, very firm. So the turgid condition of plant cell is really important. Okay, it's really important. Plant cell has to be in a turgid condition, especially for herbaceous plant. Okay, so herbaceous plant are plants that are herbs lah. So it doesn't have the hardwood, for example. Okay, herbaceous plant. So this is the normal condition for plant cell. But if you immerse the plant cell in isotonic solution, so there will be no net movement of water molecule. Water diffuse in and out at the same rate. So it causes no change in the cell's volume. Okay, so the cell, the plant cell is in the condition we call it as flaccid. Okay, so and leading to the plant to wilt. Lama-lama dia akan jadi layu, dia tak segah macam tu. Okay. So and then the next one we have if you immerse the plant cell in hypertonic solution, okay. So outside is really concentrated. So there is a net outflow of water by process of osmosis, and then the cell vacuole will shrink. Okay, and the plasma membrane pulls away from the cell wall and causes the cell to shrivel. And then this is what we call it as the process is plasmolysis. Can you replace this or you slash this with plasmolyze? Can the cell become plasmolyzed? Avoid this one. Okay, the cell becomes plasmolyzed. Okay, causes the plant to wilt. Again, it causes the plant to even more layu lah macam tu kan. So the normal solution for plant is hypotonic. Okay. So next one, we are going to look at the third type of passive transport. We have facilitated diffusion. Okay. So it is a process of solute movement across the membrane with the help of transport protein. Okay. So the movement of solute across the membrane, it requires transport protein, either channel protein or carrier protein. So the movement of this solute, okay, through the transport protein or using the transport protein, it follows concentration gradient. What does follows concentration gradient means? You can use these three terms: follows concentration gradient, down concentration gradient, or from high concentration of solute to low concentration of solute, the same thing. Okay. So by moving the solute using this transport protein, it increases the rate of diffusion, okay, across the membrane. It increases the flow of solute really fast, okay, if the solute move using the transport protein. So the transport protein are very specific on which chemicals they allows to pass through. So every transport protein is specific to the solute that it is going to move. Okay, very specific. So if water, for the movement of water, for example, it requires aquaporine. So no other molecule can use the aquaporine to move across the membrane, okay, using the aquaporine. So every transport protein is specific for the solute that it is going to move. And then two types, we have the channel protein, we have the carrier protein. So this is the channel protein because this one is passive transport. The movement of solute is down concentration gradient. So as you can see on the outside here, this in this case, for example, the solute is on the outside is higher compared to the inside. So the solute will move down its concentration gradient. Will move from high solute concentration to low solute concentration region. Okay, from extracellular fluid into the cytoplasm. So through the channel protein. For the carrier protein, so as you can see, so this carrier protein, it has binding site to bind to the specific solute. Okay, so the solute will move down its concentration gradient. Okay, so from high to low. Okay, so next, okay, so channel protein. Let's look at channel protein first. Membrane proteins that form passageways. Okay, that has a hydrophilic channel here, passageways, okay, or hydrophilic channels for selected substances. So first one, the function of the channel protein provide passageways that allow specific molecules or ions to cross the membrane. Okay, cross the membrane, it can be either the molecule move from inside to outside or from outside to inside. It depends on the situation. Okay. So the feature of this channel protein is that it has hydrophilic channel. Okay, hydrophilic channel or hydrophilic passageway. Our boleh slash kat sini kan, hydrophilic passageway or hydrophilic channel. Okay, so it allows water molecule or ions, small ions, to move quickly from one side of the membrane to the other. Okay, so example we have aquaporin channel protein for water. And then the ion channels can ions such as sodium ion, potassium ion, chloride ion can move in and out of the cell through these ion channels. Okay. So let's look at on the aquaporin first, can. So aquaporins are the water channel proteins. Okay. Facilitate the massive amounts of diffusion of water molecules in plants and also animal cells. So without aquaporin, the rate of water movement across the phospholipid bilayer is low. Without the aquaporin, water can still diffuse across the plasma membrane because water is really, really tiny molecule. It can still pass through the membrane, but the rate is very slow. So to make sure that the water will diffuse across the membrane really fast, okay, so the water will use the aquaporin. Water can still move because it is a small molecule, but at a really slow rate because water is a polar molecule. And as for the as the phospholipid bilayer is hydrophobic, it has hydrophobic region here. Okay, so water can still move, but at slow rate. So the movement of water molecule across membrane using aquaporin is also considered as osmosis. So if you look at the process here, the movement of water, if it just diffuse across the phospholipid bilayer, it is osmosis. If it uses the channel protein, it is also osmosis. Okay, asalkan pergerakan air itu daripada it's high free water concentration to low free water concentration, the movement of water, okay, is osmosis, regardless if it just diffuse or using the aquaporin. Okay, so that is the function of the aquaporin. And then okay, boleh faham So let's look at the next one, the channel protein. Okay, so ion channel. So this ion channel, we have several types. Okay, a few types. So the first type of ion channel, here we have non-gated ion channel. Kau boleh tak kat sini? Non-gated ion channel or leak ion channel. Tambah ion kat sini. Okay, ion. So ion bukan ion, ion. Okay. So water, so it says here always open. So that's why it uses the term leak. The one feature of this leak ion channel is that it is always open. Okay, it is always open and responsible for the permeability of specific types of ions. So simply allows ion to pass through the channel without any resistance, without any hindrance. Okay. So let's say for example, in this diagram here, okay, so here on the outside extracellular fluid side, okay, the concentration of sodium ion is high. Okay, compared in the cytoplasm, the concentration of sodium ion is low. Okay, so the movement of this sodium ion from outside to inside cell, it uses the leak ion channel. Okay, for example, okay. So for potassium, as for the potassium, okay, so the concentration of potassium inside cell is high. As you can see, the movement from inside to outside. So therefore, potassium inside cell is high, and then outside cell, the concentration of potassium is low. So potassium will move down its concentration gradient from inside cell with higher potassium to outside cell with lower potassium using the leak ion channel or non-gated ion channel. It is always open to allow this ion to move down the concentration gradient. Okay, so kalau potassium guna potassium punya lah, kalau sodium guna sodium punya lah kan. Yang ni dia tak boleh guna untuk potassium, tak boleh. Yang ni dia tak boleh guna untuk sodium, tak boleh kan. So every channel protein, okay, it is specific for the solute or ion that it is going to move. Okay. Okay, so tadi non-gated, it is always open. It allows the ion to move down concentration gradient. But this one here, we have the gated ion channel, still passive. Okay, still passive. Still the movement is down concentration gradient, but the channel it only opens if there is a stimulus. Okay, so can open or close in response to a stimulus. So the stimulus can be with the presence of chemical molecule. So this chemical molecule, we call it as ligand. So example is for example neurotransmitter. Okay, with the presence of neurotransmitter, it causes the gated ion channel to open. For example, nanti kita tengok example and then we have what other stimulus is electrical charge, okay, across the membrane, change in voltage across the membrane, okay, or due to pressure, mechanical, okay. So let's look at one by one. Okay, so the first one here, we have the ligand gated channel. Ligand gated channel is chemical molecule such as, for example, the presence of neurotransmitter. It says here, it open and close in response to ligand or chemical molecule binding to the receptors such as neurotransmitter. Example of neurotransmitter is acetylcholine. Okay, so as you can see, these neurotransmitters such as acetylcholine binds to the ligand gated channel here. This ligand gated channel, it has binding site where the neurotransmitter can bind. So as the neurotransmitter binds, initially the ligand gated channel will close. Okay, it is initially closed. Okay, close. Okay, so because why? Because you see there is no neurotransmitter binds. But if neurotransmitter binds to the ligand gated channel, it causes the channel to open and allow sodium to diffuse. The process is still facilitated diffusion, okay, from high to low. Awak kena tahu dekat pada sel haiwan ni kan, secara normalnya sodium is always high on the outside, okay, it is always low on the inside. So the sodium will move from high to low, okay. So if it moves using the ligand gated, it needs to open the ligand gated channel first using the neurotransmitter. Where can we observe the application of this phenomenon? So it applies to neuron cell. Okay, neuron cell. Neuron cell, it has, if you look at the structure of neuron cell, tengok lain lah. Neuron cell dia ada dendrites kan? Dendrites. So ini ada cell body dia. Dendrites yang ada this extension here. Okay. So from cell body it has axon. And then it has axon terminal. Okay. So besar sangat tapi tak apalah. On the previous neuron here, previous neuron dia ada, sorry. The neuron on the previous neuron dia ada ni lah kan. So the axon terminal of the we call it as postsynaptic neuron. Tak apa, ini yang awak belajar nanti. The neuron that is transmitting electrical impulse. Okay. So what is electrical impulse actually? It is due to the movement of ion in and out of the cell. The ion involved is sodium ion and also potassium ion. Okay. So in order for the sodium ion to enter into the neuron that is receiving the electrical impulse, on the dendrite of that cell, okay, dendrite, it has the ligand gated channel. Ligand gated channel. So initially the ligand gated channel closed. So when it closes, how can the sodium ion enters? Okay. So this postsynaptic neuron kan, kita belajar kat sini neuron ni dia ada vesicles that releases neurotransmitter. Okay, neurotransmitter is released. This area we call it as a synaptic cleft. Okay, synaptic cleft. So neurotransmitter will be released into the synaptic cleft. The neurotransmitter will bind to this ligand-gated channel, causes the channel to open, causes the sodium ion to be able to enter. That is due to the presence of ligand. Okay, so that is ligand gated. Kalau yang kedua tadi apa? Voltage gated channel. Okay, the stimulus is electrical. Electrical. So the stimulus is electrical or voltage. Change in voltage. Okay, change in voltage. So we have here voltage gated channel. Okay, the change is due to electrical change. Okay. So where can we find the ligand gated channel? Ligand gated channel is located along the axon. Okay, along the axon. Okay, along the axon of a neuron. Along the axon of neuron is where actually the electrical impulse is going to be generated. Okay, so how to allow this voltage gated channel to open? It is due to the change in electrical impulse. So the normal condition for a cell to be in is inside the cell it has to be negatively charged, normal. Okay, but neuron, it is a special cell that is able to generate electrical impulse. What is electrical impulse? The voltage inside the membrane, inside the axon, is supposed to change from negative, it becomes positive. Okay, it becomes positive. So how to make inside the cell to become positive? Can inside the cell it has to be positive, so it opens the voltage gated channel. So voltage gated channel opens. When it opens, it allows sodium ion to enter. Okay, so when sodium is positively charged, okay, sodium is positively charged. So as sodium enters, it changes the voltage from negative becomes positive. Okay, so it opens more voltage gated channel. So more sodium enters. So that is how a neuron generate electrical impulse. Due to the opening of voltage gated channel, sodium enters. Okay, it changes the voltage inside cell from negative become positive. Okay. Okay. So this one can. So voltage gated channel open or close in response to small changes. Okay. Small voltage changes. For example, voltage gated sodium ion channel. Okay. So for this diagram here, initially inside the cell it is negatively charged. Okay. Outside it is positively charged. Sebab apa saya cakap tadi, sodium outside cell is always high. Okay, is always high. So but if there's presence of voltage gated channel, okay, when the voltage across membrane changes from negative to positive, it causes the channel to open. Okay. In this case sepatutnya sodium tu masuk, tapi tak apalah. Kan nampak? Sepatutnya sodium masuk. Oh buat macam ni. Sodium ni sepatutnya masuk Okay. Voltage gated. Sodium supposed to enter into the cell. You refer to this diagram here. Do not refer to this one. Okay. Sodium supposed to enter into the cell. Okay. So that is voltage gated. And then we have the third one. Mechanically gated open in response to physical deformation. So if you apply pressure, so it allows the channel to open. Okay, so response to physical deformation of the receptor, as in sensory receptor of touch and pressure, such as in your skin, in your ear, kan? So if there's physical deformation, it causes the channel to open. Okay, so this slide here is the same as this slide. Okay, it is the same. But for this one, you have to make sure you refer to this one. Okay, sodium enters due to the opening of voltage gated channel for sodium. Itu kalau untuk sodium. Kalau voltage gated channel for potassium, potassium keluar. Okay. So same slide. Okay. So three types of gated ion channel, ligand gated. Okay. So example here we have acetylcholine. Acetylcholine is a type of chemical molecule. Ligand. It is a type of neurotransmitter. So initially, the ligand gated channel close. But if the acetylcholine binds to the receptor, it causes the ligand gated channel to open. Sodium enters into the cell. Okay, so acetylcholine receptor found at the neuromuscular junction. And what is neuromuscular junction? The neuron is transmitting electrical impulse to a muscle cell. Okay, so as impulse from this neuron is accepted by the muscle cell, again, it causes the muscle cell to be able to contract. Okay, so it only happens if in the presence of acetylcholine opening the ligand gated channel, causing sodium to enter into the cell. A voltage gated channel open or close in response to changes in membrane potential. Okay, what is membrane potential? Difference in electrical charge inside and outside cell. Inside, usually it is negatively charged. Outside, usually it is positively charged. Because we are looking at a neuron cell, at the axon region, there will be the presence of voltage-gated sodium ion channel that opens, okay, if the membrane changes its electrical charge from negative become positive. So it opens the voltage-gated channel to allow more sodium to enter and causes the generation of electrical impulse in that neuron. Okay, so for the third one, okay, so we have here the mechanically gated channel. Okay, so open and close in response to physical force, pressure, stretch, or vibration. Okay, channels in touch receptors of skin and hair cells in the inner ear. Okay, so these are examples of gated ion channel: ligand gated, voltage gated, and mechanically gated. Okay, so let's look at this disease. Okay, we have so far. Do you have any question regarding the ion channel, the gated one, the non-gated, gated? If no, I'm going to proceed. Okay. Okay, so next one we have this disease, cystic fibrosis. Okay, so cystic fibrosis is a type of genetic disorder. It is genetically inherited. Okay, so cystic fibrosis is a disease that causes thick, sticky mucus to build up in the lungs, digestive tract, and other areas of the body. This is the feature of the disease. Okay, so what causes the disease? It is due to mutation to a certain gene. Okay, so a gene in your DNA, it code for the synthesis of specific protein. Our nucleus, it contains all your genetic informations, okay, all your genes. So all your genes code for the synthesis of specific protein. Okay, in this case, the gene that is supposed to make protein that is going to become chloride ion channel is mutated. It is mutated. The coding has changed. So when the coding changed, it causes the synthesis of the channel protein to be affected. Okay. So either the channel protein cannot be synthesized, so it causes the channel to be absent, or it is synthesized but do not function, defective. Okay. So the channel here is the chloride ion channel. Okay, chloride ion channel proteins. Okay, chloride ion channel. Okay. So when it is defective or when it is absent, okay, so the channel cannot move chloride ion across the cell. Okay, so chloride ion channel is important to regulate the flow of water and salt across membranes of epithelial cells. Okay, so how? Nanti kita tengok sekejap lagi ya. So without the chloride ion channel, without the ion channel here, it leads to build up of mucus in trachea, lungs, pancreas, liver, gallbladder, reproductive organ, sweat glands, and other organs. Okay, so let's look at the mechanism. Okay, so here we have the cells. Okay, the cells, let's say the cells lining the trachea. So we know that the trachea is where it is your airways, that when you inhale, air will enter through the trachea and it enters into your lung for gas exchange. So the air that you breathe in is it is not clean, it is full of contaminant. So the cell lining your trachea, there will be a type of cell, we call it as the goblet cells. The function of these goblet cells is to secrete mucus. The function of the mucus initially is to trap the contaminant. Okay, but if there's a defect in the chloride ion, it causes the mucus to be thickened, to build up. Okay, so when the mucus thicken, build up, the cell line in the trachea, as you can see, it has cilia. Okay, so initially the mucus that traps the contaminant, the cilia was supposed to swipe the mucus with all the contaminant. Either you will cough it out, or you engulf, so or you swallow. Okay, swallow yeah, put through your esophagus and down into your stomach, so your stomach acid will digest lah. But if there's mutation to this channel protein, the chloride ion cannot be transported across the cell. Okay, so in the lungs, the chloride ion channel protein transport chloride ion from inside to outside of the cell. This is in the normal situation, in normal condition for healthy person. So when chloride inside cell move from inside to outside of the cell, so the presence of the chloride ion outside cell will attract water. Okay, it attracts water and causes the mucus to be diluted. Dia jadi tak pekat lah. Okay. So extracellular chloride is required to maintain healthy mucus as it attracts water. So mucus yang terhasil tu tak adalah jadi pekat, dia akan dicairkan. So bila cair, bolehlah mucus tu digerakkan oleh cilia. Okay, so if there's mutation to this chloride ion channel, okay, so the chloride ion cannot be transported out and causes the chloride ion to be accumulated inside cell. So there will be high chloride inside cell, okay, and less chloride outside cell. So when there's less chloride outside cell, less chloride ion outside cell, so less water is attracted to the area that produces the mucus. And so the mucus become thicker and stickier than normal. Okay, so sebab apa? Chloride tak boleh nak dibawa keluar daripada sel, tak boleh nak attract air. Okay, so less water is being secreted into the mucus outside of the epithelial cells. So when there's no water due to less chloride or no chloride, so it causes the mucus to thick and cannot be swiped up by the cilia. So the mucus initially it is to trap pathogen, contaminants, but if the mucus build up, all those contaminants, all those viruses, pathogen will stay in your respiratory tract. Kan? So it causes infection, kan? So leads to persistent lung infection, chronic bronchitis, and also pneumonia. Okay, so this is the chloride ion channel for the normal healthy person. Kan? So supposedly the chloride ion channel is supposed to transport chloride from inside cell to outside cell. Okay, inside cell there is high concentration of chloride ion, so it moves to the outside with low concentration of the chloride. So this movement of chloride ion is important to attract water so that the mucus cannot thicken. Okay, so this is normal CFTR, the name for the chloride ion channel. Carilah dia punya abbreviation ni stand for what, kan? CFTR. Okay, so normal CFTR channel, ataupun kalau tak nak ingat nama, just chloride ion channel, itu je. Okay, so so this is the normal channel. Okay, so the chloride ion move out of the cell, attract water to the mucus, keep mucus thin and healthy. But if there's mutation to the channel, okay, so mutant channel does not move chloride ion, causing sticky mucus to build up in outside of the mucus, outside of the cell. Okay, so chloride ion stays inside cell, less water enters into the mucus become thick and sticky. Okay, So this is where this is another diagram lah. As you can see, the build up of mucus in the alveoli in your lung, or mucus that blocks the pancreatic ducts, okay, in your digestive system as well, right? So you can scan this QR if you want to watch more information on cystic fibrosis lah. Okay. So next one we have for the carrier protein. Okay, so for tadi this one is channel protein. Okay, channel protein. So example of disease involved is cystic fibrosis. So now let's look at carrier protein. Carrier protein, it can involve in both passive and active transport. If passive, it is still facilitated diffusion. Okay, so small hydrophilic molecules such as glucose, amino acids cross the cell using the carrier protein. Okay, so this process is facilitated diffusion because, as you can see from this diagram here, this carrier protein, it moves glucose outside cell with higher concentration of glucose outside. So glucose high on the outside binds to the binding site of this carrier protein, causes the carrier protein to change shape, and glucose will release into the cells. So these are the keywords that you need to pay attention lah, kan? So the solutes or the glucose in this case bind to the specific binding site. This is the keyword that you need to highlight. Okay, you need to know how to explain. So the solute binds to specific binding site. Bukan active site. Active site is for enzyme. Binding site on the carrier protein. So the carrier protein changes shape. Okay, so the molecule is released on the other side of the membrane. In this case, inside cell. Okay, so because the solute move from high to low, so the process is spontaneous. It doesn't require energy, especially energy from ATP. Okay, no energy input is required. So this is the example. Glucose binds. Glucose is on outside of the cell. So as the glucose bind, it causes the carrier protein to change shape. So glucose is released into cell. So example here we have glucose transporter. So this is the animation. Outside cell, there will be high concentration of glucose. Glucose bind to the binding site, causes the channel protein the carrier protein to change shape, and the solute enters into the cell. Okay. Okay, so another genetic disease here we have is cystinuria. Okay, tadi cystic fibrosis. Now we have cystinuria, another type of genetic disease, inherited disease. Okay, so what is the cause? So it is caused by too much amino acid cysteine in the urine. Okay, so normally cysteine entering kidney cells is absorbed back into the bloodstream. You have your kidney. Okay, the kidney is an organ, so it is a pair of organ that is function to filter your blood. Okay, your blood will be filtered by the kidney. Inside the kidney, there will be tiny units. Okay, many tiny units. We call it as the nephron. Okay, so we have the glomerulus, the structure of nephron lah. You have the glomerulus, and then surrounding the glomerulus, you have the Bowman capsule. Okay, and then it extends to proximal tubule, this loop of Henle, can descending and then ascending, and then distal tubule, and then collecting ducts. And so as blood is filtered in the glomerulus area, so the fluid from the blood will enter into the Bowman capsule. Okay, becomes filtrate. Okay, so this filtrate, it has a lot of nutrients. It has a lot of water. Okay, so these nutrients, including the amino acid cysteine, for example, that enters in the filtrate, has to be reabsorbed back in your blood. Okay, but the carrier protein that is supposed to transport cysteine from the filtrate in the nephron back into your blood, it is not functioning well, or it is absent, it is defective. So instead of the amino acid absorbed back into your blood, it maintains in your nephrons. Okay, it maintains in your nephrons. It accumulates. Okay, and then also it accumulates in the fluid that will become your urine. When the cysteine accumulate, it becomes stone or crystals that will get stuck in your kidney, or it gets stuck in your urinary bladder, or it gets stuck in your ureter. Okay, So the cysteine accumulates in urine and may form crystal or stones, block kidney, ureter, or urinary bladder. Okay, so this is for the normal carrier protein. Suppose this is the, for example, this is inside a nephron. Okay, inside a nephron where it has filtrate that contains cysteine. So this filtrate later will become urine. Okay, so suppose this cysteine has to be reabsorbed by into your blood vessels, enters into your blood circulation. So the transportation of cysteine is using the carrier protein. This is for the normal healthy cells, okay, within the kidney. But for those that have cystinuria, this carrier protein, okay, it is either defective or absent, missing, okay, defective or absent. So what happens is that the cysteine will accumulate in your kidney, okay, accumulate in the fluid that will become your filtrate, your urine, can accumulate and become crystal and stone. So it gets stuck in the kidney, in the urinary bladder, as you can see, for example, here, kan. So this is the structure of nephrons, kan. So you have the formation of kidney stones in the kidney, or stuck in the ureter, or stuck in the urinary bladder. Okay, so this is the symptom for having kidney stones: stomach pain, backache, vomiting, dizziness, you have fever, kan, blood in the urine. So obviously your urine should not contain blood. Okay, so if your urine contains blood, so it means that there's something wrong with the kidney. Okay, so you have to do some checkup. Okay, so that is for the cystinuria. So you have cystic fibrosis, you have cystinuria due to the missing or defective transport protein. Okay, do you have any question before we enter into the active transport? Okay. Okay, so let's tak apa, kita rehat sekejap 2 minit. Okay, so for you to digest apa on that before we move on into the active transport. Okay, so let's continue on active transport. Okay, so this is the definition for active transport. It is the process of ions or molecules movement across the cell membrane aided by a transport protein against the concentration gradient. What is against concentration gradient? From low solute concentration to high solute concentration. Okay. So, so this process only requires carrier protein. It doesn't require channel protein. Channel protein is for facilitated diffusion only. Okay. So because the movement of solute is against their concentration gradient, so the process requires energy, okay, by ATP molecules. Okay, so ATP molecules are produced from the process of cellular respiration. So cells that carry out active transport have high respiratory rates. Okay, so therefore to produce a lot of ATP, the cell will need a lot of mitochondria. And so a lot of mitochondria to carry out a lot of cellular respiration to produce a lot of ATP to provide energy for the process of active transport. So enables a cell to maintain an internal concentration of small solute that differ from concentration in its environment. And so in this case, for example, these cells needs to maintain this solute low and high outside cell. Low inside cell, high outside the cell. Okay. In that example lah, in this example kan. So next, okay. So we have here kan, so a type of transport protein that involve in active transport. We call this as electrogenic pump. Okay, what is pump? Pump here is referring to transport protein that transport solutes against concentration gradient using energy from ATP. That is pump. It is a type of carrier protein that transports solute using energy from ATP. Okay. Okay. So so voltage, can what is voltage? Voltage is created by differences in the distribution of positive and negative ions across a membrane. Previously, I have mentioned to you that for a cell, in the normal condition, inside the cell it has to be negatively charged. Okay. Outside the cell, it has to be positively charged. So the difference of charge inside and outside cell, we call this as membrane potential. Okay, the difference in the charge, it creates membrane potential that can be measured in voltage. Okay, membrane potential is the voltage across membrane, negative inside, positive outside, usually. Okay, but for neuron, in order for a neuron to change from negative to positive, can it also makes put the voltage to change, can. So that creates um, that allows a neuron to be able to generate electrical impulse daripada negatif jadi positif. Macam tu lah. Okay, so that is membrane potential. Boleh negatif ataupun boleh positif as long as you have difference in the charge inside and outside cell. So an electrogenic pump is a transport protein or carrier protein that generate a voltage across a membrane. So we have two type of electrogenic pump. The first one we have here for animal cell will have sodium potassium pump. For the plant cells or fungi or bacteria, they have proton pump. Okay, proton pump is hydrogen ion. Okay, proton pump. So let's look at the sodium potassium pump first. Okay, so this is sodium potassium pump. As you can see, so this pump here is they are actually carrier protein. They have binding site to bind to the solute, that is solutes lah, solute in this case ions, to move across membrane against concentration gradient. So bendanya adalah pump. Okay, so this the transport protein, we call it as pump. The process of moving, okay, the ions, okay, dia guna words yang sama kan, so it is also pumps. Okay, pumps. Okay, so the 3 sodium ion out of the cell for every 2 potassium that enters into the cell. Jadi awak kena ingat kan, so this is the plasma membrane of the cell. This is on the outside cell, this is on the inside cell. Tadi saya mention dekat luar sel ni, outside cell, sodium ion is always high. Inside cell, the sodium ion is always low. Okay, because this is active transport, okay, the ion will move from low concentration to high concentration against concentration gradient. So in this case, because the sodium potassium pump will pump free sodium out, can from inside to outside cell, it requires the energy input from ATP. So sodium inside cell will bind to the binding site. Okay, so this will trigger ATP to come. Okay, so this ATP will cause the phosphorylation of the sodium potassium pump. So ATP is adenosine triphosphate. Okay, so the ATP will release one of its phosphate through the process of hydrolysis. Okay, ATP hydrolyzes its terminal phosphate, and the phosphate will be added to this sodium potassium pump. Okay, the process we call it as phosphorylation. Sodium binding stimulates phosphorylation by ATP. Okay, so when this happens, it causes the sodium potassium pump to change its shape. Okay, so when the sodium potassium pump changes its shape, it causes the sodium ion to be released to the outside of the cell. Okay, so when this happens, okay, so you see that the binding site for potassium now exposed to the outside. Okay, so it causes the potassium ion now to bind to the binding site. You look at on this, the first diagram here, the concentration of potassium outside is low. It needs to enter into the cell with high potassium ion concentration from low to high against concentration gradient. Okay, so that's why the process requires energy. So as this happens, when the potassium ion binds, okay, it causes this phosphate to be released. Okay, so the release of this phosphate, it causes the sodium potassium pump to change back to its original shape. Or boleh tulis kat sini original shape or conformation. Slash kat sini shape. Senang. Guna shape je. Changes to its original shape. Okay. So when the sodium potassium pump changes back to its original shape, potassium will enter into the cell. Okay. So I give you 2 minutes for you to digest the process. If you have questions, so please ask. 2 minutes. You look at the animation. Awak punya gambar, nanti awak photostat semua dan PDF tak ada animation macam ni. Record lah dengan awak punya telefon ke apa. Sama je. Okay. Sebab dalam sel, low sodium, sodium ion binds to the binding site. So datang ATP, berlaku hydrolysis. The terminal phosphate is released and attached to the sodium potassium pump. It causes the sodium potassium pump to change, release sodium outside. So it releases the binding site for potassium. Potassium from outside binds to the binding site. Release phosphate. Protein changes back to its original shape, releasing potassium into the cell. Itu je.
Miss,
Yeah.
so what is the function of the electrogenic pump like the sodium potassium pump in the body?
This one, the function we are going to look at next in our slide. Okay, next, the application. Boleh?
Okay,
Okay.
thank you.
Tapi selalunya, okay, easy example, dia tak ada easy. Maksudnya, okay, ini apply pada neuron juga. Okay. Hari ni kita akan belajar dia, proses ni kita akan belajar dengan lagi detail lah semester depan. Dalam sel neuron ni, okay, neuron ni bila dia dalam keadaan dia tak menghasilkan any electrical impulse, dia duduk diam, dia resting state. Okay, so this resting state dia nak make sure dalam neuron tu charge dia negatif 70. Okay, negatif 70 disebabkan oleh nampak sodium kat dalam ni rendah tapi potassium tinggi. Macam mana nak maintain this concentration difference ni dengan adanya sodium potassium pump. Dengan adanya sodium potassium pump ni, it maintains inside the neuron ni to have negative 70 millivolt dengan adanya sodium potassium pump. Tapi macam mana neuron ni boleh menghasilkan action potential? Sebab neuron ni dia special. Dia ada yang tadi, voltage gated ion channel. Bila ada voltage gated ion channel, kan, dia akan menyebabkan sodium masuk dalam daripada negatif jadi positif. Boleh hasilkan electrical impulse. Tapi bila neuron tu tak hasilkan any impulse, dia dalam keadaan resting ni akan dimaintainkan balik semula oleh sodium potassium pump. Nampak tak statement kat sini? Ni. Enables a cell to maintain an internal concentration of small solute that differ from concentration in its environment. Contohnya ada pada neuron. Neuron yang dalam keadaan resting kena dalam satu negatif 70 dan dimaintainkan oleh salah satu faktornya adalah sodium potassium pump. Okay. So another application kita tengok nanti. Okay. So next one we have the proton pump. Okay. So proton pump for plant cell. Okay. Bacterial cell, fungi. Okay. Proton is referring to the hydrogen ions. Okay. So it is a pump. Ingat pump ni adalah transport protein yang transport solute against concentration gradient menggunakan energi daripada ATP. That is pump. Okay. So a pump that translocates positive charge in the form of hydrogen ion, proton. Okay. So actively transport hydrogen ion, which is proton, out of the cell. Okay, that is the feature of the proton pump. Sodium from inside low concentration to outside high concentration. It is active transport. Sodium low, bukan sodium, hydrogen ion. Okay, hydrogen ion outside high. Okay, from low to high is active transport. Okay, it requires energy from ATP. So the voltage and the hydrogen ion concentration or gradient, okay, hydrogen ion concentration gradient, saya sebut ni hydrogen ion concentration gradient, okay, ataupun electrochemical gradient, represents a dual energy source that can drive other processes such as the uptake of nutrients. Okay, kita tengok application dia sekejap lagi. Okay, so cumanya awak kena tahu the proton pump, it pumps hydrogen ion from inside cell with low hydrogen ion concentration. It pumps out, get to the outside of the cell where there's high concentration of hydrogen ion. So it requires energy from ATP. So the application kita tengok ni. It involves in the uptake of nutrients. So let's look at that slide here. The uptake of the nutrients. Okay. So from proton pump, it leads to co-transport process. Okay. A co-transport system moves solute across a membrane by indirect active transport. Two different solutes are transported at the same time. So, kita tengok dulu dia punya explanation. So the movement of one solute, okay, which is in this case here hydrogen ion, inside cell, down its concentration gradient from inside to outside cell, from low concentration to high concentration, down its concentration gradient, provides an energy. Okay, nampak? Dual energy tadi kan? Dia kata provides an energy for transport of another solute, in this case, for example, nutrients such as glucose or sucrose, up its concentration gradient. Okay, so however, an energy source such as ATP is required to power the pump, okay, the proton pump or the sodium potassium pump that produces the concentration gradient, high concentration of certain ion outside cell. Dia kata dual kan? Dual energy. Satu energy, the first energy it comes from, okay, it comes from the the accumulation of hydrogen ion outside. Okay. Nampak ni? The movement of solute such as hydrogen ion down its concentration gradient provide the first form of energy. Okay. The accumulation of hydrogen ion ni dia macam jadi sumber potential energy. Okay. Untuk bawa apa? Untuk bawa sucrose daripada luar sel masuk ke dalam. Sucrose get up its concentration gradient. Sucrose luar sel rendah. Okay. Dalam sel tinggi. Again, kan? From low to high of that nutrients. From low to high is active transport. Cumanya sucrose ni bila dia nak bawa masuk ke dalam daripada rendah ke tinggi, daripada luar sel ke dalam sel, active transport kan, tapi awak nampak this transport protein doesn't require energy from ATP, but it uses the energy from the accumulation of hydrogen ion outside cell. Okay, so once there you have a high concentration of hydrogen ion outside cell, what happen is that the hydrogen ion will diffuse back into the cell, diffuse back from high to low. Okay, so the process of hydrogen ion diffusing back into the cell, it causes another form of transport protein. We call it as co-transporter. Okay, these co-transporters allow hydrogen ion to diffuse back, and at the same time, it provides energy for this co-transporter to move sucrose from outside into the cell. Dia macam potential energy lah. Okay, hydrogen ion diffuse masuk, that force, okay, it causes sucrose to also enter using the co-transporter into the cell. So in order for the sucrose to enter into the cell, it enters into the cell with hydrogen ion together, okay, or entering using the co-transporter. Nampak ni? Okay. So it says here, however, an energy source such as ATP is required to power the pump, the proton pump, that produces the concentration gradient or high concentration of the hydrogen ion outside cell. So the process of transporting sucrose indirectly, okay, it requires energy from ATP. It is a form of active transport. Walaupun tak nampak, dekat co-transport ni ada menggunakan energy daripada ATP, sebab ATP tu digunakan initially by the proton pump. Okay. Tak apa, nanti kita tengok sekejap lagi. Ada lagi the detailed explanation. Okay, so this one is for plant cell. Okay. Plant cell, it carries out photosynthesis, it produces sugar, from sugar glucose, fructose, it produces sucrose. Sucrose will be taken into the plant cells such as to be stored in the fruits of that plant. Okay, so that you have to accumulate sucrose in the fruit. Yeah, so the process is co-transport. Okay, so this one, plus for this one is sodium potassium pump kan. So sodium potassium pump is for animal cell. Okay, animal cell. So as you can see, this one here, it involves the sodium potassium pump to pump sodium out, kan? 3 sodium to 2 potassium. 3 sodium out and 2 potassium in. So the purpose is so that you accumulate sodium ion outside cell. Okay, so when there's a high concentration of sodium ion outside cell, glucose is also outside cell. Okay, so glucose, how does it enters? It uses the co-transport system. So glucose will enter together with sodium ion, and this is a form of active transport. Okay, the accumulation of sodium ion provides a form of energy to transport sucrose into the cell using the co-transporter. This is co-transporter. The name of this co-transporter is sodium ion glucose uh, transporter. Boleh guna kat sini kalau senang kan, sodium ion glucose co-transporter je. Okay. So okay, let's look at the process lah kan. So from here lead to this slide here. If you have question after this one, okay. So co-transport is a system of indirect active transport. Okay. So indirect active transport. So jap. So for plant, it involves the proton pump. Okay. For animal, it involves the sodium, potassium pump. So these two pumps, it requires energy input from ATP. Okay. So, nampak ni? Co-transport is a system of indirect active transport. Okay, where for animal cell it involves the proton pump, animal sodium potassium pump. So two different solute are transported at the same time. Two different solutes. What is the solute? Okay, so for if the plant cell, okay, it uses the co-transporter to transport hydrogen ion along with sucrose. Okay, if it's animal cell, the sodium ion along with glucose. Okay, so there will be accumulation of this ion, hydrogen ion and sodium ion outside cell. Okay, it is a form of energy to bring in nutrients into cell. Okay, so one solute, hydrogen ion and sodium ion, move down its concentration gradient. Move down its concentration gradient if you have the sodium ion or the hydrogen ion accumulating outside. Okay, accumulating outside due to these two pumps. So its energy helps another solute such as sucrose or glucose to move against its concentration gradient. Okay, so the nutrient outside is low, that you want to accumulate inside cell high, from low to high. Okay, so ATP is needed indirectly to power a proton pump that creates the ion gradient. Example, hydrogen ion sucrose transport in plant. Okay, sodium ion sodium ion glucose transport in animal. Okay, in animal. Okay, so let's look at on the on plant first. Okay, in plant, so hydrogen ion sucrose co-transport. Ini dia. Okay, so this is in plant. Hydrogen ion sucrose co-transport in animal. Sodium ion glucose co-transport. So let's look at the mechanism, okay, for the plant cell. Okay. Example, hydrogen ion sucrose co-transporter. Co-transporter is the name of the transport protein. The process is co-transport. Okay. The process is co-transport. The name of the transport protein is co-transporter. In this case, hydrogen ion sucrose co-transporter that occurs in plants. Okay. So this co-transporter is to transport sucrose produced by photosynthesis into cells in the veins of leaves. Okay. So the process again. So you want to accumulate sucrose in the vein of leaf. The leaf carry out the photosynthesis, okay, produces the sugar, but the vein is to transport the sugar produced to other parts of the plants, kan? So to other parts. So dia kena accumulate dulu, bawa glucose yang terhasil tu ke other parts of the plant. Sucrose yang terhasil tu to other parts of the plant. Okay. So in order to accumulate the vein with sucrose, it uses hydrogen ion co-transporter. So let's look at the process. Okay, so the process. First, it uses the proton pump. Okay, proton pump uses energy from ATP to pump. Okay, to pump hydrogen ion from inside cell to the outside of the cell. Inside cell, the concentration of hydrogen ion is low. Outside cell, the concentration of hydrogen ion is high. Okay, from low to high. So that's why it requires energy from ATP. Okay, so once the proton pump pumps the hydrogen ion from inside to outside cell, there will be an accumulation of hydrogen ion outside. Okay, so this is a form of energy. We call this energy as proton motive force. Okay, create a form of energy, we call it as proton motive force. Okay, so hydrogen ion accumulate outside cell. So what happened next is that the second transport protein will involve, which is the hydrogen ion sucrose co-transporter. So this co-transporter brings sucrose from outside cell. The concentration of sucrose outside cell is low. You want to accumulate the sucrose in the vein. So in the vein, there will be high concentration of sucrose from low to high. It is a form of active transport but does not directly use energy from ATP. But it uses the diffusion of hydrogen ion from outside into cell. Okay, so it says here, the hydrogen ion sucrose co-transporter brings sucrose into the cell against its concentration gradient from low to high. Okay, together with hydrogen ion that diffuses back in, okay, down its concentration gradient from high to low. The movement of hydrogen ion from outside to cell is facilitated diffusion, but the movement of sucrose is active transport. Okay. Right. So the hydrogen ion, so this hydrogen ion gradient generated by the proton ATP powered pump drives the active transport of sucrose and other nutrients into the plant. So co-transporter is a form of active transport where the co-transporter indirectly uses energy from ATP. Okay. Question? 2 minutes. I give you time for you to digest. Sebab kalau tak ada faham ni, yang sodium potassium pump lagi dia apa require a lot more understanding compared to this one. So fahamkan ni dulu. Kalau ada soalan tanya. Orang nampak macam ada arrow sini kan? Buang arrow tu, arrow dia hanya masuk ke dalam saja. Okay. Arrow hydrogen ion masuk ke dalam. Hydrogen ion, proses yang masuk tu adalah diffusion. Ya, soalan? Apa soalan tu?
Ada.
Ya.
Proton multifast tu yang sebabkan sukus tu boleh masuk ke dalam sel ke?
Proton, kalau tak ada proton accumulate dekat luar ni, sucrose tak boleh masuk.
Mungkin ada.
Okay. Sucrose hanya boleh masuk dalam sel sebab sucrose ni kat luar rendah, kat dalam tinggi. Dia perlukan tenaga. Tapi tenaga tu bukan daripada ATP. Tenaga tu datang daripada pergerakan hydrogen ion ni yang dah accumulate kat luar dengan banyaknya. Kan? So, bila hydrogen ion dah accumulate dengan banyaknya kat luar, so hydrogen ion akan diffuse balik masuk ke dalam. Pergerakan hydrogen ion tu menyediakan satu form of tenaga untuk bawa masuk sucrose ke dalam sel. Ni, this point here. This hydrogen ion gradient, high outside, okay, generated by the proton pump, ATP powered pump ni, okay, drive the active transport of sucrose and other nutrients into the plant cell. That's why I cakap tadi dual energy. One from ATP, one from the proton gradient. Okay, dual energy. One from ATP, one from the proton gradient. Okay, so let's move on to the next one, to the next part. Okay, ini a little bit more complicated, but okay, still okay. Okay, untuk awak fahamkan. Okay, a bit more complicated than the proton pump implant. This one it applies for animal cell. Okay, for animal, for human lah, kan? So it says here. So it uses this one, sodium ion glucose co-transporter. So you look at here, this is a cell, this is cells lining your small intestine. Kita tahu fungsi intestine awak untuk digestion and absorption of nutrients. Okay, lumen intestine tu, the cell lining the intestine, we call it as epithelial cells. Belajar kan dulu, epithelial cells ni dia ada microvilli. So that's why dia punya, awak ada nampak kat sini, sel dia tu ada extension macam ni. These are microvilli. Okay, so this is cell lining the small intestine. This is the lumen of small intestine. Awak makan, awak minum, semua nutrients semua ada dalam lumen of the intestine. Okay, this is the epithelial cells lining the lumen of intestine. Lumen of your intestine ni, kenapa dia boleh berlakunya absorption of nutrients? Sebelah small intestine tu ada your blood capillaries, your blood vessels. Okay, so daripada nutrients, daripada lumen yang awak makan, air yang awak minum, so daripada lumen masuk ke dalam your blood circulation in your blood capillaries. This is the blood capillaries containing your blood with the water, with the glucose, the nutrients that is going to be transported throughout your body. Okay, so the epithelial cells lining the lumen of intestine here, okay, lumen, blood vessels, you have several types of transport protein. So the first one here, we have the aquaporins. Awak minum air, masuk dalam lumen intestine. So through the aquaporins, water will be reabsorbed, and then through the process of another diffusion of water, kan, through the aquaporins, water enter into your blood vessels. Okay, and then we have, kan, so ini yang satu. Dia ada aquaporin. So another transport protein here we have the sodium ion glucose co-transporter. Okay, sodium ion glucose co-transporter. Awak makan ada garam, awak makan ada gula kan, daripada nasi awak makan semua tu kan. So this one sodium ion glucose co-transporter helps to transport the sodium into the cell and the glucose also into the cell. Okay, glucose and sodium enters into the cell using the co-transporter. Without the sodium, glucose cannot enter. Okay. So yang ketiga kita ada sodium potassium pump. Okay. Sodium potassium pumps, what it does is that it pumps sodium out and potassium in. Sodium enters into your blood. Okay. So yang keempat, tadi glucose dah bawa masuk dalam sel kan? So you have the glucose transport protein. Okay, the name is GLUT2 transport protein. Will transport the glucose in the epithelial cell into your, so that glucose enter into your blood. Okay, so this is your blood capillaries. Okay, your blood capillaries containing your blood. Okay. Alright. So kan ada empat jenis transport protein yang awak kena fahamkan. Note kat situ. The aquaporins, the sodium ion glucose co-transporter, the sodium potassium pump, and also the glucose transport protein. Awak tak payah ingat GLUT2 pun tak apa, just ingat dia adalah glucose transport protein. GLUT2 transport protein is glucose transport protein. Okay. So, the application of this knowledge untuk apa? Kita tengok to treat dehydration due to diarrhea or vomiting, patients are given oral rehydration salt (ORS). Or ceret-beret, contohnya lepas makan makanan yang dah basi whatever, ada bacteria. Okay, so this bacteria it has toxin, and then the toxin will block these aquaporins. Okay, block these aquaporins. So bila awak makan, awak minum, the toxin from the bacteria will block, and then the nutrients, the water will not be absorbed. Dia akan keluar as ceret-beret lah. Okay. So to treat this, the doctor will give you the salts, the air garam lah kan ya dalam paket-paket tu, oral rehydration salts. So the solution contains a high concentration of salt and glucose. Ya, awak minum dia rasa manis-manis masin macam tu kan. So the process of water reabsorption involves several transport protein. Kita habiskan slide ni, nanti kita sambung So sodium ion glucose co-transporter present in plasma membrane of epithelial cells of sodium ion glucose co-transporter, this one here, present in the plasma membrane of epithelial cells. Okay. Plasma membrane of epithelial cells of the small intestine, the brush border. This is brush border. Okay, macrovilli tu. So the apical surface. This is the apical surface. Apical surface are the one that is facing the lumen of small intestine. Awak minum, semua masuk dalam lumen. So the apical surface will have this sodium ion glucose co-transporter. Okay. So next we have the sodium ion potassium pump. Here. Yang ini dia dekat dengan blood vessels. Okay. So sodium ion potassium pump and glucose transport protein, which is GLUT2 transport protein, located on the basolateral surface, bahagian bawah yang dekat dengan blood vessels. Okay. And then we have the aquaporins. Aquaporin dua-dua side. The apical ada, the basolateral pun ada. Okay. For absorption of water. Aquaporin facilitate the movement of water from lumen, enter into the epithelial cells, from the epithelial cells enter into your blood through the aquaporins. Okay. Okay. So saya nak untuk next lecture, sebelum kita masuk next lecture hari Jumaat, Isnin Jumaat ya, awak punya lecture ni Kan? Baca ni dulu, proses ni dulu, baca. Okay. Baca, baru kita, baru faham proses tu dengan lebih jelas. Sebelum masuk dalam lecture hari Jumaat, tolong baca dulu. Okay, tolong baca dulu. Okay, so kita habis. Okay, so sebelum kita habis lecture kita, ada soalan ke? Tak ada. So sebelum habis saya nak remind untuk hari Rabu punya lab session kan adalah consultation untuk S10. Nanti saya keluarkan jadual lah untuk awak masuk dalam ni untuk kita discuss awak punya lab by group. Maksudnya kumpulan 1 masuk dulu, lepas tu kumpulan 2, tak adalah semua masuk serentak. Okay? Itu dia. Okay class, so thank you. S9 you maintain untuk tutorial.
Thank you, madam.
Okay.
Thank you, madam.
Thank you, madam.
Okay. Okay, sama.